Hey friends, we'll talk about viruses today. So viruses called bacteriophages, kind of example of viruses, they infect the bacteria named E. coli. Okay, so these viruses which infect the bacteria, for example, E. coli, are known as bacteriophages, and these viruses they live a kind of borrowed life between the life forms and the chemicals. Okay, this is a structure of a bacteriophage which infects the bacteria E. coli, right? So this purple color is the host, and the small hexagonal head is the bacteriophage, right? So a virus consists of a nucleic acid surrounded by a protein coat. Outside is a protein coat, inside which is a nucleic acid. And the nucleic acid can be DNA or it can be RNA. It can be single stranded RNA, it can be single stranded DNA, or it can be double stranded also. So discovery of viruses. So first, tobacco mosaic disease, which shows stunted growth in the tobacco plants. It gives a mosaic coloration. Okay, this disease shows the stunted growth, and what it gives a mosaic coloration to the plant, to the tobacco plant. Okay, and in the late 1800s, it was found that these are uh, particles which are smaller than the bacteria, and they cause the disease. Okay, they are smaller than the bacteria. Next is in it will be in 1935. Okay, so there was a scientist named Wendell Stanley, 1935. Okay, so there was a scientist named Wendell Stanley. He confirmed that this crystallizing the infectious particle, now which is known as TMV. Okay, so see how it he has done that. He has extracted the sap from tobacco plant, with is having the disease tobacco mosaic disease. This extract has been passed through a porcelain filter, which is used to trap the bacteria, and this is then extract is then rubbed on healthy tobacco plants, and it shows. Yes, and the you can see this is the healthy tobacco plant, and this healthy tobacco plant is when exposed to the extract, it becomes infected. That means this extract had some virus particles, right? So this was proved by Wendell Stanley, right? This contains tobacco mosaic virus. So how is the structure of the virus? Okay, how is the structure of the virus? So these viruses, first of all, they are not cells, they are non-living. Okay, outside the host cell, they are non-living, but when they enter the host cell, they become living. As already discussed, it has a protein coat and inside is the viral genome. And the viral genome can be double-stranded or single-stranded DNA, double-stranded or single-stranded RNA. Depending on it, can be a DNA virus or it can be RNA virus, right? So nucleic acid is present inside the viral head. Head is made up of protein coat. Okay, coming to the protein coat, capsid and envelope. So capsid is a protein shell that encloses the viral genome. Okay, the head part is the capsid. Basically, in case of bacteriophage, and this protein subunits are known as capsomere. These are the subunits which make the protein coat, right? So capsid can have various structures as you can see here. So first one shows kind of helical symmetry, and this second one shows kind of icosahedral symmetry. Okay, helical symmetry is in case of TMV, then icosahedral symmetry where it is found. This is found in T4 virus. Okay, so the last one is bacteriophage having DNA, tail sheet, tail fiber, and the tail fiber goes and attaches itself to the bacteria. So capsid, which is enclosing the viral genome, and there are RNA virus also, DNA virus also. Inside is the nucleic acid, which you can see. There are membranous envelope onto which glycoprotein is attached. Got it? So example is second one icosahedral is also in adenovirus, Third one is influenza virus where glycoprotein is there, right? So first one is what? Helical symmetry. Second is icosahedral, which is example is T4 virus, adenovirus. So you can see the structure, the shape. Okay, so it's a kind of polyhedron with 20 equilateral triangle faces. This is helical symmetry. You can see inside which is the nucleic acid. So example is tobacco mosaic virus. The third one is influenza virus where outside is the glycoprotein which acts as a ligand and it attaches to the host cell uh, on the receptor which is in the host cell. Next is the bacteriophage where you saw that the upper part is the head part which have the nucleic acid, then it is the collar part, then it is a sheath, then it is a tail fiber. Okay. So some viruses they have the membranous envelope that help them to infect the host. Okay. And this envelope surrounds the capsid. So first is the genome, nuclear genome, then it's the capsid, then it's the envelope. Okay, for example, in case of influenza virus and some other virus found in animals. So viral envelopes which are derived. So example I write here, one example where viral envelopes are found is influenza virus. Okay. Next is viral envelopes which are derived from host cell membrane. Okay, so where are they derived from? Host cell membrane. Okay, it consists of what? It consists of viral and host cell molecules. Both molecules are present here in viral envelope. Basically, it is derived from host cell membrane. Next, we come to bacteriophages, also called as phages, and they are the viruses which infect the bacteria. So, one example, name a va- name a virus which infect bacteria. It's bacteriophage. Okay. Now, the bacteriophages, the important characteristics of it is what? It is the most complex capsid. It has the most complex capsid and found among the viruses. And they have an elongated capsid head, the hexagonal head, as I said, inside which the DNA is present, and a protein tail piece. Okay, as I showed tail sheath and tail fiber that attaches first to the host. Which is the host here? Host is bacteria and injects a fudge inside. Okay, this helps the fudge to inject its DNA inside the host. Next is viruses reproduce only in the host cell. So they are non-living outside the host cell, but inside the host cell they become living, and they are obligate intracellular parasite. Okay. So, obligate means compulsorily intracellular parasite, which means they can reproduce only within the host cell. And they have a host range. Okay, they have a host range and a limited number of host cells that it can infect. All cannot infect the all of the host. Okay. So next we come to what? So what we discussed right now, we discussed that they have a limited host range. Okay. So next we move forward. What? So till now we remember that they are obligate intracellular parasite. Okay. So what are the features now? So what are the important features of the viral reproduction? So I write here one by one. The important one is they enter the host cell. Okay, they enter the host cell. How? They inject their nucleic acid into the host cell. Okay, they inject the nucleic acid into the host cell and they synthesize the proteins inside the host cell, the viral proteins inside the host cell by using the host machinery. Host enzymes, host ribosomes, host tRNAs. So everything they use from the host, right? Amino acids also. Okay, and ATP and other things also. So after they use the host machinery, some of the nucleic acid remains in the mRNA form, and some they form the proteins, and after which there will be the assembly of new viral particles or new viruses, which will break the host cell and it will come out. Okay, so when they break when they release from the host cell by breaking the host cell membrane spatially, that is called as lysis. Okay, so nucleic acid capsomeres are formed and the new viruses are released. So this is the process of the lytic cycle. What happens? The first step is attachment. Okay, first step they attaches. So entry and uncoating. After attachment, there is entry and uncoating where they release their nuclear particles into the host cell. And you can see that the outside part remains outside the host cell. Nucleic acid comes in. So what happens? It multiplies in manifold. Some will form the protein and some will remain in the mRNA form, and the protein part will then enclose this mRNA part, and this capsid proteins and viral DNA, they assemble and form the new viral particles which release outside the host membrane by breaking or by lysing the host cell. So these phages, they are the best understood of all viruses and have two reproductive mechanisms, the lytic cycle and the lysogenic cycle. Clear? Lytic cycle, what are the features of the lytic cycle? Why the bacteria uses lytic cycle and when? Okay, and when they uses the lysogenic cycle, and like what are the features? So lytic cycle shows the death of the host cell. Okay, it shows the death of the host cell, and the next part is the next important feature of the lytic cycle is it digests the host cell membrane. It breaks or digests the host cell wall, sorry, and the new particles, the new fudges are released outside of the host membrane. Okay, so first step was like attachment. Okay, the second was what? Release, like penetration of the DNA into the host cell. The third step is multiplication of the uh, genome, and then these lytic cycle, the fudges which shows this lytic cycle only the fudges which exhibits the lytic cycle only, lytic cycle, are known as virulent bacteriophages or simply virulent fudges. Okay, so bacteriophages, uh, as I said, they shows lytic and lysogenic cycle, and basically lytic one are virulent fudges, and the bacterial defense system. Okay, so when these bacteriophages like infect the uh, host, like bacteria, the bacteria have their own defense mechanism. Okay, bacteria have restriction enzymes, which is known as restriction digestion enzymes. These enzymes, what do? They digest the viral DNA. Okay, they digest the viral DNA. They can recognize the viral DNA and digest them, but bacterial genome remains uninfected. Okay, they will not infect the bacterial genome. Okay, so they can recognize only the viral particles or viral DNA and digest them for their own defense. Okay, we isolate this restriction enzymes and we use for several lab purposes. So first step is the attachment. Second step is the penetration or entry of the DNA. Attachment, then is the penetration, which is the entry of the viral DNA into the host, that is the bacteria. The third step is the digestion or breaking of the host DNA into fragments. Okay, it will be degraded, host DNA will be degraded. Then the next step will be the synthesis of virus nucleic acid and protein. Virus nucleic acid can be DNA or RNA depending on which, since it is a bacteriophage, it will be DNA. So synthesis of virus nucleic acid and protein, then the nucleic acid and protein will assemble and it will be packaged, and new fudge will be released outside the host cell by lysis. by breaking the host cell wall. Okay, since it's a bacteria, it will break the host cell wall. Got it? So moving forward to the next part. Yes, so it will be released outside by breaking the host cell wall. Yes, this is the pink colored part is the host cell wall.